Okay, thanks, Nina. Welcome, everyone. It's my great pleasure to invite Andries van der Meer to give us his talk on organ-on-a-chip devices for vascular research. I'm going to introduce him now. So Andries is an associate professor at the Faculty of Science and Technology at the University of Twente in the Netherlands, and he is the lead of the organ-on-chips research team and part of the Applied Stem Cell Technologies group of the Bioengineering Technologies cluster. He's supervising currently 6 PhD students and coordinating multiple national and European research projects on the development and application of organ-on-chip devices. Before joining the University of Twente as a faculty in 2015, Dr. van der Meer worked as a senior research fellow at Harvard Medical School and the Wyss Institute for Biologically Inspired Engineering at Harvard University. He actively developed organ-on-chip device models for the blood-brain barrier and the alveolus for the Defense Advanced Research Project Agency in DARPA. And this part of the microphysiological system program coordinator and collaborative projects between the Wyss Institute organ-on-chip startup company, Emulate, and Janssen Pharmaceuticals as well. Before joining Harvard, he was a postdoctoral fellow at Professor Albert van der Berg's BIOS Lab on a Chip group at the University of Twente in the Netherlands. And during that time, he also served as an assistant coordinator for the project Beyond Borders, Organs on Chips, of the Dutch Royal Academy. This project led to the founding of the Dutch Human Organ and Disease Model Technologies Organ on Chip Consortium, for which Dr. van der Meer is currently the university representative. He obtained his PhD biomedical engineering from the University of Twente in the Netherlands in 2010 and an MSc prior to that from the University of Groningen in the Netherlands as well in 2005. So without further ado, it's my great pleasure to welcome Andries to give us his talk, Organ on Chip Devices for Vascular Research. Over to you, Andries.
Thank you. Thank you very much, Tim. Is my screen already sharing? Can people see stuff? I would like to thank you all for inviting me, for turning up in such great numbers, for also still having the opportunity now to present this work in front of this community, because I was really excited actually to come to the UK and in response to your invitation, Tim, but unfortunately it couldn't be. Still, I'm now very happy that that at least I get to show some of this work. So I'll be talking about the work that I'm currently coordinating, and I focus a lot in my lab on the development of what we call blood vessels on chips. I'm very much a, let's say, applied scientist or translational scientist. So I really enjoyed also this morning the more fundamental biomedical and physiological talks. My background is also in biology, medical biology. So yeah, I've been away for a while, but I always enjoy coming back to these types of meetings. So like I mentioned, I have a very strong translational attitude, and we do work a lot also with pharmaceutical companies. And the reason for that is that, the preclinical drug development in pharmaceutical industry actually is not that efficient, to put it mildly. So the numbers here speak for themselves. So 80% of the current therapeutic drug candidates that are brought to clinical trials, typically already in phase 2, phase 3 clinical trials, fail. And 40% of all these drug candidates, they fail because they don't show any efficacy. So basically, after all the preclinical optimization testing, including all the assays in vitro that we know, all the animal models, still it turns out that actually these drugs simply don't work the first time they're being taken into human. And you can imagine, of course, with the costs related to clinical trials, that pharmaceutical industry doesn't take drugs into clinical trials very lightly, but apparently the evidence simply is not strong enough to avoid these types of failures. And I think also rather surprisingly is that 15% of all drug candidates still fail in the clinic also because of unexpected toxicity. So it's been, again, extensively tested in animals, typically also already in phase 1 clinical trials in healthy human volunteers. But it turns out that if you bring them into the patient groups, a lot of them, of course, are not healthy volunteers. Obviously, they do have comorbidities, they do have aging, they do have overall health problems, and that's why we see actually a lot of unexpected toxicity still in these drug candidates. So basically, this means that there is a niche there for scientists like myself to collaborate with the pharmaceutical industry to improve this preclinical testing in drug development. So this is what is, let's say, the motivation for the work that we do in the lab. And like I said, we work mostly on vessels on a chip. And this started actually already quite a long time ago, over, let's say, 15 years ago. So these are the resulting publications of that work, in which we basically realized that devices as the one that we see here, known as microfluidic devices, can be used as sort of microbioreactors. So we can use them as an in vitro model in which we culture human cells and then expose them to very defined culture conditions. And then in particular, of course, if we think about vascular biology, these types of devices that basically contain little microchannels, they of course are highly advantageous because you can imagine we have a way here of applying defined flow profiles. We have thus ways of applying defined shear stress profiles. And as you can see here, endothelial cells and F-actin staining, they very strongly react, of course, to shear stress, as most of you will know. But there's even, let's say, more advanced little tricks that you can do here, for example, by doing selective wounding and looking at wound healing also under shear stress, or looking at wound healing under controlled gradients, etc. So initially, this was, let's say, a research field in its own right, where people focused a lot on doing cell biology in these microbioreactors, microfluidic chips, as we call them, because a lot of the fabrication methods of how we make these devices in the lab is actually derived from how we normally do this with microelectronics. So thus the term chips comes in. What I will show you in let's say the coming 15 minutes or so is two aspects of where we have been going since. So one of them focuses a lot on the perfusion of these types of systems with blood, which is something that we've been focusing on for the past 5 years or so, and is becoming very interesting as a research direction in its own right. The other aspect that, of course, I want to talk about a lot with you is, okay, this is fairly simple systems, as we can see here on this slide. But about 10 years ago, we realized that we can actually consider these blood vessels on chips a building block, as it were, for more complex in vitro disease models and tissue models. This is then known in the field as organs on chips. And I will show you how we actually take what we develop here and then integrate that into progressively more complex and physiologically complex models. Okay, so I'll start with, let's say, the part about blood perfusion in these devices. And just as a quick disclaimer here, so we do perfuse here with human whole blood. Typically, a typical experiment, also what I will show you, is about 30 to 40 minutes. And at that point, we start running into problems. Not so much problems actually in this part here in the chip. So this is where the microchannel is, that is lined with endothelial cells in this case. But typically we run into problems here, either in the reservoir or in the pump. So we have a syringe pump that withdraws the blood from this reservoir through the channel, etc. So typically 40-45 minutes into an experiment, you will see coagulation, clotting, and the flow will start to decline. So this is the main reason why these experiments that we are currently doing are relatively short term. So let's zoom in on those experiments a little bit, and I wanted to very briefly touch on what we currently use as the vessel wall tissue. So I showed you in the previous slide that we line these chips with vascular endothelium. And here we see another microscopy, confocal fluorescence microscopy reconstruction of one of these chips. In this case, lined by human umbilical vein endothelial cells and then stained for weak adherin, and of course nuclei in blue. And what we have been doing and are doing is actually exploring which cell types can we integrate in these types of chips. Because, well, as you can see here, we basically, just for a sense of scale, this is just under a millimeter, what we see here in width. And as you can see, basically what we end up with is what you would consider a blood vessel, but then with a rectangular lumen. And that comes with all kinds of problems and challenges, and I will show you also in the coming few slides. But we have, of course, full control over the geometry of these chips. We have control over which cells do we see in there, so we can do either human umbilical vein endothelial cells or the induced pluripotent stem cell derived endothelial cells. We will collaborate a lot with the laboratory of Christine Mummery at the Leiden University Medical Center to integrate these types of cells. And then in addition, we have, of course, ways to pre-activate or treat these endothelial cells. So we can either culture them under controlled conditions or start to treat them with, in this case, increasing concentrations of TNF-alpha from 5 nanogram per mil to up to 100 nanogram per milliliter. And then we see by ICAM1 expression, in this case, a typical endothelial activation marker, that we can tweak or tune the response of this lining of the microchannel. And this is also, I think, important if we think about the current COVID-19 crisis, of course, is that we start to realize by now that a lot of the serious cases and people suffering from the serious COVID-19 cases and showing very severe side effects typically have a lot of problems, not just with airway, but actually have a problem mostly with vasculature and with thrombosis and cardiovascular complications. And that means that, so currently what we're trying to do is to take this type of concept where we activate the endothelial lining, but then instead of just using a recombinant TNF-alpha, we take, for example, serum from COVID-19 patients and see if we can stratify them based on severity. Because presumably, of course, some of these thrombotic side effects are due to the cytokine storm, as we define that in these patients. So the more cytokines are circulating, we hope we can pick some of that up in these relatively simple models. So now that we at least I've given you a little bit more background of what these channels look like and what the cell culture in these channels looks like. Let's dive into the blood perfusion. So here we see another confocal microscopy reconstruction of one of these channels. And as you can see, we basically have tested 3 conditions here. So either we do not line the microchannel with vascular endothelium, but we only leave it functionalized with collagen 1. Or we do culture endothelium in there, but do not activate it, or we have activated the endothelium with TNF-alpha. And then we perfuse with blood, and the fluorescent signal that we see here is basically the blood platelets as labeled with a non-blocking CD41 fluorescently labeled antibody. And you'll have to believe me, but this video here in the center is also running, but yeah, of course, like normally, if we consider the physiological situation, normal human endothelium should, of course, not activate platelets, let alone stimulate aggregation. However, if we have pre-activated the endothelium with TNF-alpha, we do see platelet adhesion, activation, aggregation. And as you can see, those patterns are actually quite distinct from the more punctuated pattern that you would see typically on collagen 1. And these types of patterns on the activated endothelium can actually become quite aggressive. So here we see an example of the end of a 30-minute run. We see here this microchannel and an endothelial nuclei labeled in blue. And then again, the platelets here labeled in red. And then everything here is decorated now with the fibrin. So in this case, we added a tiny bit of fluorescently labeled fibrinogen to the blood sample just before perfusion. And this soluble fibrinogen actually gets incorporated in fibrin networks that we see here. Another maybe remark on the side is already that I just showed you that we have a sort of rectangular cross-section microchannel, and you can already see here that does have effect. So we have most of the fibrin formation actually on the sides of the channel. And the reason for this is that you basically have little corners with almost no flow. It's almost dead volume there. So you have stagnant blood sitting here in these corners, and that leads to fibrin formation. So these typical patterns that we see here, you would also see in the normal untreated, unactivated endothelium. Interestingly, of course, I just pointed you to the typical pattern that we see. So the aggregation of these platelets, and now we also see the fibrin, so the decoration with fibrin here. These are typical patterns that you would also find in animal models. So here's an example of a laser injury model in the mouse, and then the platelets in this case are labeled here in green. So initially we have platelet adhesion aggregation sitting on the wound, and then that gets decorated and stabilized in red here with that fibrin. And that's the type of pattern that actually we see also in these vessels on chips. So I of course started my presentation by saying that, yeah, we like to be translational and at least find collaborations with pharmaceutical industry. So in order to sort of convince them that this is a model that has added value, we did a number of drug studies, and basically we took two approaches there. So one approach actually is quite straightforward. So what we see here is the control situation. So control channel, meaning endothelial cell lining, but not activated, still perfused with blood. But as you can see, you hardly get any platelet adhesion or fibrin formation in that case. Then if the endothelium has been pre-activated with TNF-alpha, as we see here, then this is the pattern that I basically just showed you. So aggregates of platelets decorated by fibrin. And then if we add drugs that are used currently in the clinic for mostly acute myocardial infarctions to quickly alleviate thrombotic load, then one of these drugs is, for example, eptifibatide, or typically marketed as integralin, a small molecule inhibitor against GP2B3A, so a receptor on the platelets. Then basically we see that this whole pattern of platelet aggregation goes away. And we see something similar with a drug that targets the same receptor, but this is a Fab fragment based, so a biological drug given intravenously typically. And there too, we can very nicely see a dose response curve. So we see here the platelet coverage in the chip. So basically the strength and the aggressiveness of this pattern. And we see that we can actually take that down almost to control levels again. So that's one approach to do it. Of course, the nice thing about perfusing these models with human blood is that you don't necessarily need to take blood from healthy volunteers, as I've shown you up until this point. We simply use a volunteer donor service. But you could, of course, also take blood from people that are already on antiplatelet medication. So yeah, you all might have heard, of course, of people that are taking low-dose aspirin after having a scare of a heart attack or an actual heart attack. And there are similar drugs that these people are taking, like clopidogrel, typically labeled as Plavix. And if we take the blood from these people on antiplatelet medication, we did a very brief exploratory study there with one of the hospitals in Boston, where we showed that blood of healthy donors still gives this kind of aggressive pattern, whereas blood of people that have been taking their antiplatelet medication, so their aspirin or Plavix, already see a very strong inhibition of that response, not down to this level of control as is in the highest concentration of the, for example, epciximab or eptifibatide, but still we can at least show efficacy of those types of drugs. So we went on to actually take this model and looked at some thrombotic side effects and thrombotic toxicity together with Janssen Pharmaceuticals. Unfortunately, I don't have time in this very brief presentation to go into that project or in that study. But yeah, if you'd like to learn more about that, that's all being published actually in this particular paper here. So you might want to look that up for, let's say, more of a relevant case study. This was more, let's say, just proof of concept, of course. But the case study that we published there, was more, let's say, cutting edge when it came to experimental pharmacological research. Okay, so then before we move on to, let's say, the organ-on-chip part of the talk, I very briefly wanted to show you and highlight one additional advantage of using these microchannels or micro-engineered chips. And the fact is, we focused a lot currently on the endothelial lining and the cell biology of it all, basically. But we know, of course, from experience, and I know I'm talking to the microcirculation community here, so and this is a whole different universe and different size range. But if we think about large artery diseases, and mostly atherosclerosis, we know that it's not simply a biological ordeal. It's also a geometrical, a flow dynamic problem in the vessels. And what we have, of course, if we look at atherosclerotic plaques, is that we typically have stenotic geometries. And it's not really known if they contribute a lot to the thrombosis that then in the end, upon plaque rupture or plaque erosion, causes heart attacks, or if this is simply a side effect of the chronic inflammatory process that's going on there. So we basically made a very simple model of a typical atherosclerotic artery by taking our microchannel and then introducing this stenotic section in there. And we ran the exact same experiment that I just showed you. And basically, so this is lined again with endothelial cells and then perfused with human whole blood. And this is then pseudo-colored in green for the highest fluorescence intensity. And then we see that basically the little aggregates still form as we see them here upstream. So basically, this is the pattern that I just showed you also in that single full screen slide. But we see here that directly downstream of this stenotic geometry, the aggregation and the clot formation actually is heavily exacerbated. And the main reason for this, and again, if you would like some more details, then by all means look up the publication. But the main reason for this is that we have a unique flow environment here that is strongly activating of the circulating von Willebrand factor. Von Willebrand factor is the #1 protein complex that under high shear stress activates and recruits platelets, and it needs to get activated. And it's particularly in this sudden onset of increased forward flow direction that we get far more VWF activated. So I think it also highlights that, yeah, if we model this ourselves and if we engineer these models ourselves, we sort of open, of course, also the possibilities of designing these types of studies instead of just only focusing on commercially available systems. So as I mentioned, this I think is also a nice bridge to organs on chips, because well, this was all the work, and we have a lot more ongoing work on blood vessels on chips. But what we have realized over the past 10 years is that actually we can use that same approach of, let's say, controlled engineering and integrating biological tissues in controlled microchips can be used to slowly also build more complex models. And yeah, for those of you that are not very familiar with this field, I think the fastest way to get you up to speed is to introduce the Long on a Chip paper. If you want the full paper, it's published here in 2010 in Science by Dan Huck, currently at UPenn, and Don Inger at the Wyss Institute in Boston. And they basically had a very simple idea. They said, let's look at the lung here, the human lung, and zoom in on the smallest functional unit of a lung, the alveolus. And let's sort of put on an engineering mindset and think about, so what do we see there? What do we need to build if we would want to build a functional unit like this? So they said, okay, well, we have an epithelial and endothelial interface, obviously, for the gas exchange. We have an air-liquid interface, and as you can see here, with each breathing motion, we actually have a strong mechanical strain on these alveoli. And they went ahead and basically took one of those models that I just showed you only for a blood vessel on chip and started to make more complex devices that then captured these aspects. So both the epithelium, the endothelium, and the breathing motion. And I have a short video, and I'm not sure if that will come off nicely in Zoom, but to basically show you schematically what this device looks like. So if we zoom in, then basically we see that internally it has 4 compartments, a blue, a red, and 2 gray ones. And the middle 2 compartments are separated by this porous, flexible membrane. So it's an elastic membrane. And they established a co-culture here of epithelial cells and endothelial cells. And then basically the two compartments here on the side are not used for cell culture, but if you apply vacuum with a cyclic pattern to the side channels, you can basically stimulate also the co-culture with this cyclic strain pattern. And then of course the top can be filled with air, and this bottom, in their study they filled it up with medium, but it is supposed of course to represent the capillaries, so it is actually nice to fill that up with blood or perfuse it with blood. And that's exactly what we did. So if we now zoom in on this red compartment here in the top, that basically, of course, is one of these blood vessels on chips, right? So basically what we have done is to take this little compartment and then integrate it back into a more complex device. So what that allows are studies for looking at epithelial endothelial crosstalk, for example. So I'm not going to go in all the details of this particular study. Again, if you want to look that up, you can look it up there. But basically what we did in this study is we stimulated the epithelium with LTS, which is a stimulus derived from a gram-negative bacterium, to sort of simulate bacterial pneumonia. And then we focused a lot on the endothelium to see, okay, we dose something in this air compartment, but how does the endothelial compartment, so this red compartment, how does that react? So we see, in that case, increased ICAM1 expression. We see increased gaps, as you can see here. So basically, the endothelial monolayer is disrupted. We see also that being accompanied by increased permeability of the endothelium, as you can see here, simply by doing dextran tracer assays. And also we see specific increased cytokine excretion. Particularly here, this big bar is IL-6, interleukin-6, interleukin-8, and MCP-1. So that allows you to very systematically actually integrate blood vessels in other and combine them with other tissues. And that's something, let's say, a theme that you can of course apply to the lung, but you could also apply that to other tissues. And we just saw, of course, a very interesting presentation about the endocardium and the myocardium. But also, if we look at the microvasculature of the heart, of course, we know there's a lot of interesting aspects there to be studied. So we basically can apply this exact same concept, say, okay, we have a blood vessel on chip, but we integrate this with a 3D cardiac tissue. So what we use to build those models with cardiac tissues is this human iPS-derived cardiomyocytes. So this is typically how they look in a 2D culture. So they are nicely contracting, but completely disorganized. And then we use these cells to basically pattern them, and just for a sense of scale, this is about 3 millimeters, pattern little cardiac tissues, as you see here, that then like to contract rhythmically, and we see some patterns and also deformation here in the center, where it contracts in a cyclic fashion. And the channel that we see running underneath here is basically the blood vessel on chip that I showed you multiple times in this presentation now. So this allows us to very systematically look at interaction between 3D cardiac tissues, consisting mostly in this case of cardiomyocytes from iPS, and then endothelial cells in the bottom channel. So that basically brings me to the end of the presentation. I see that I need to speed up also. I just want to very briefly touch upon the fact that I do realize that a lot of what I've shown is all, let's say, basically two-dimensional. It's basically flat lining endothelium lining rectangular channels. There are ways of going more three-dimensional. Some of that is, for example, very similar to the fibrin assay that we saw earlier this morning, where you embed endothelial cells in fibrin in one of these devices, and they will form a network themselves that eventually becomes perfusible. But I think what is important also to realize is that we need ways to make vessels and vascular networks that are also very well controlled, because that's the whole concept, of course, of making these in vitro models, these on-chip models, is that we are the ones as researchers that have the full control. So one of these ways is basically to, so I'm going to skip how we make this, but one of these ways is to take one of these channels, fill it fully with a collagen 1 gel, as schematically given here. And then we have a way to basically form a very nice circular lumen in the center of the collagen 1 gel. And this is on the right-hand side, the end result of this. And then this allows us then to basically line this with endothelium, as you see here. But then also the nice thing is that because collagen 1, of course, is a soft and a biocompatible material, you can establish co-cultures between pericytes and endothelial cells, as shown here. And in this case, between astrocytes and endothelial cells, as shown here, because this was mostly in the context of building a blood-brain barrier on chip model. And then the 3D reconstructions of these types of vessels look more or less like this. So we have endothelial lining here on the left-hand side, and the endothelial astrocyte co-culture here on the right-hand side. And the nice thing is, because it's all contained in this microfluidic channel, we can still start building complexity on there. So we can still separate it with a flexible membrane. We can still put another compartment on top of there. So I think this is the whole concept of what we're trying to do, is to go from this organ on chip in a very systematic and controlled way to try and, well, yeah, we're a bunch of nerds at the University of Twente after all. So everybody's trying to push and see where are the limits. Well, yeah, we basically believe that there are no limits there. If you maintain this level of control, that you can actually set up multi-organ models, right? So, and there's a lot of studies out there that, for example, already show interactions between liver and gut on chip models, or models with cancer and looking at metastasis to the different tissues. So this seems a little bit like science fiction, but actually, yeah, we're really trying to work our way towards this by applying this concept of controlled engineering. With that, I'm sorry I went a little bit over time, but thanks a lot. Thanks again for the kind invitation and the kind introduction. And I hope to also attend the second part. I'm looking forward also to the rest of the presentations.
Thank you, Andries. That's absolutely perfect. There's lots of questions. I'm going to put a couple of technical ones first, put them together. We have a question from Giovanni on sensitivity of the endothelium to oxygen and the permeability of the PDMS. I'm sure that's the kind of question. And also associated with that as well, the stiffness of the PDMS and how that helps. And obviously when you're starting to mix with the hydrogels, that's another order again, which is absolutely fantastic. We're trying to go down that way as well. So maybe just a couple of words on those two things, oxygen and stiffness, and how that affects the endothelium.
Yeah, so the easiest one is the stiffness. So the formulation of PDMS, it's a silicone rubbery material, obviously, but the stiffness itself is super physiological. So the endothelial cells, and this has been studied, they don't see the difference between polystyrene, for example, and this PDMS. So you do need gels like collagen 1 or other hydrogels, synthetic or biological, to get closer to what they feel in vivo. The oxygen part, PDMS is highly oxygen permeable. So, and that's a big advantage, and actually has kickstarted this field very nicely, in the sense that it's a very forgiving material. If you have a lot of cells sitting in a very small volume, as in the systems that I showed you, then it's actually nice that you can still put them statically in an incubator, and they at least will get enough oxygen and gas exchange. However, we are now beginning to see that this is problematic, particularly since we're getting more and more interested in, for example, ischemia reperfusion, where we want very clear-cut control over the oxygen levels. And then, yeah, PDMS is simply not the material of choice anymore. So we move, for example, to other materials, and some of those are quite common materials like PMMA, so plexiglass type materials.
Yeah. There's a question about the lining of the endothelium, and some endothelium do line up under flow and so on, and maybe this is talking about the oxygen permeability of the length of exposures. Obviously the clotting that happens relatively soon, 45 minutes is something, but how long can they last? Also questions on TNF. Have you tried TGF-beta and looked at von Willebrand's expression? And just to follow up on that as well, is it possible to multi-layer? So, for example, adding pericytes into these kind of systems?
Yeah, so again, to start with the last one, yes, in principle, it's possible also to do pericytes and pericyte endothelial systems, but then, yeah, you do need the soft hydrogel. So the easy way actually to do this is to make sure that first you fill the channel with the collagen 1 hydrogel, you make a lumen. So there's let's say, tricks to make an open lumen in this hydrogel. and then first seed the parasites and then seed endothelial cells. So it's a very simple protocol in a way. You just simply do sequential seeding. But what that leads to is basically that the parasites, they feel at home seeing endothelial cells and the basal membrane on top of them, and they're okay with collagen directly underneath them. So you can still get a fine monolayer of endothelial cells in that sense. So some of those other questions, for example, the TGF-beta, we haven't looked into stimulation with TGF-beta. So we have done some studies with TGF-beta inhibition, and that does affect, for example, aspects of the network in other chips that I very briefly touched upon. But no, I don't have anything. For the VWF, we did, like in that PNAS study that I very briefly showed, we did a lot of staining in that case. So also showing indeed that the endothelial cells are VWF positive, but also showing that these stenotic regions actually lead to local accumulation of VWF because all these VWF chains get entangled in a way.
Yeah. Just one last one, if we can. Yeah, we've used interferon gamma compared to TNF, and we see clotting as well. We see platelet clots with that, just as we go through. And finally, from Lily, what advantage do you think your technique has over using ex vivo techniques such as dissecting out an intact pectinate muscle strip, for example?
Yeah, very good point. And generally, actually, smooth muscle cell incorporation is not trivial here. So parasite integration in these models is easier. Smooth muscle cell and realistic smooth muscle cell integration is actually quite tricky. So, but the nice thing, and we are collaborating with people, particularly at the Erasmus Medical Center in Rotterdam, they have a lot of experience with this, with dissecting and then ex vivo testing of larger blood vessels. And then looking at dilation, etc. The big advantage, if you can manage to still realistically build such a bigger blood vessel, including the smooth muscle, in one of these chips, is that you are completely free to choose. So you choose in terms of cell material mainly. So we are actually working with these people in Rotterdam to use human iPS-derived endothelial cells and human iPS-derived smooth muscle cells. Some of those have been genetically modified, and then integrate those together into these larger models. And some of them we want to just keep in the chip and look at. Some of them are actually taken out of the chip and then tested on their typical ex vivo setups. So I think there's a lot of synergy there to be had. Yeah, the big advantage of course, this technology is that it can be based on human cell material.
Excellent. I think we'll have to call time on that. That's absolutely perfect. On behalf of the BMS, thank you very much, Andries. If you'd like to go and see the hundreds of questions that are also on there, lots to do with the recruitment of inflammatory cells and things like that, then please do take a look. And that's it for me. I'm going to hand over to Jackie Watmore, who will take us through the final 4 talks. Thank you very much.